Okay, so that started. So let me see if this one is like a full screen. Is it okay? Can you see the full screen? Yeah. So
Yes.
so I was in somewhere here, I think. Right. This is what I was doing last. Talked about receptor and accordance, and this one. So let me just start back up by just one slide, right? And then from here I will move forward. So what we talked about in the last class, where we left off, was somewhere here, where we talked about a ligand and receptor, and what we said was that because the cell is enclosed in a lipid bilayer, you have resistance to molecules traversing in and out. And there are different categories of molecules. For example, if you have just oxygen, carbon dioxide, they just permeate through. If you have an organic molecule like benzene, that will go through. If you have water and ethanol, which are on the smaller polar molecules, then they can also pass through. But the moment you get into larger polar molecules, or ionic molecules, or ions rather, you have a problem. And then you need to have something that allows it to go through. And for large polar molecules, you can have some kind of a channel to allow it to go through. And if you have molecules just slightly smaller than that, if you have ions of this kind, then you can have gated channels for these ions. And the most famous of these are the potassium-sodium channels, which are there on neurons, as well as calcium channels. Of course, hydrogen ions are usually part of these sympoters and antipoters. So also with chlorine ions. But we'll talk a little bit about the calcium ion today, and that will happen just in a couple of slides from now. And I talked about this as an imperfect analogy, where essentially the receptor ligand kind of binding allows things to happen inside the cell. And here also you see that someone has to kind of pass through the gate by giving a boarding pass before you can really board the flight and get on with your life, that kind of thing. So we are still in that phase where we need to talk about these things. So if you can look at this slide here, and I did already talk about these channels, but what we have not talked about in some detail is the transporters that you have for these glucose molecules, and the glucose PTH system is well known. And glucose, mannose, and galactose, I think, or I'm not sure what galactose, glucose and mannose for sure are part of this. And they would essentially have some kind of mediators. And you can have two cycles of these mediators which allow the selection of either glucose or mannose to enter into the cell. So the PTH system is quite active, and the reason why this is important is because this is the first step of the glycolytic pathway. So whatever goes in is phosphorylated. So you get glucose becomes hexose phosphate, and it's getting, and there are other mechanisms by which this internalization takes place, and one of them is something that is very commonly understood in viruses, or observed in viruses, for example, endocytosis, where what happens is the proteins on the surface of the vesicle, and in this case the virus vesicle, will essentially merge with the bilayer of the cell and then enter into the cell. So there are various mechanisms that are there, and the details of this are quite fascinating. And to understand some of this has been a very serious study in virus-host interactions, and exactly how this happens is still well under. I mean, is being researched by many groups across the world. And what you find is that there are asymmetries on the surface of the otherwise absolutely symmetrical surface of viruses, and those asymmetries begin to occur when this endocytosis takes place. And along the edges of asymmetry, this virus will then break open and release the active material into the cell. And we talked about this also a little bit, where we talked about the secondary messengers in particular. And usually what will happen is, the secondary messengers that are there will be acting to bring about certain actions within the cell. So the response is this one, which is the secondary messengers, and the initiator is the ligand binding to the receptor. So that's the signal that is there, which is the reason why we call this, signal transduction or signal pathways. And from here you have an amplifying enzyme. So usually, it depends on what is the molecule that is going to be the secondary metabolite and what is this ligand receptor here that is going to couple with this or mediate some changes in this enzyme. Just give me a second. Sorry. So you can see that, from the previous slide, here. So this amplifying enzyme is something that is that's something that could be as simple as converting ATP to cyclic AMP, for example. And otherwise, you can have calcium also as secondary messengers here. So there are two different routes through which primarily these things happen. If this cyclic AMP, most likely this is going to be a GTPase protein. And otherwise, you'll have usually another group which will be going through phospholipase C mediated action. Anyway, I talked about this a bit. Usually, it's because of the structural changes that this binding takes place. Here, you have dimerization takes place, and that allows for this phosphorylation. So this binding will allow a dimerization, and that dimerization will allow this phosphorylation to take place. Once it is phosphorylated, it's like it got some power to do some kind of an action. And so when you have this here, you can see that phospholipase C is, for example, or phosphinol 3 kinase is going to be the other group. So there are various kinds of groups that are there, which takes into account what will be the secondary messengers downstream of this. I will not go into these things. I think I've talked about this a bit. I think I kind of moved in too early on the slides. So this also we talked about. I just remember now. So just to recall this thing. So you can see that if you have adrenaline as a ligand, and then you have this receptor binding to this location here, then this alpha, beta, gamma units of this GTP protein is going to be modified by actually coming in association with this receptor. And then that will essentially give you this beta gamma will separate out, and the alpha sub unit will essentially work with adenylyl cyclase to give you cyclic AMP that we talked about earlier. And as you can see, there is a certain amount of selectivity that is there, and this is something that has to be kept in mind. So there is a tenfold excess of GTP over GDP in the cell. So because of that, there's a driving that takes place to give you excess of cyclic AMP. All right. So I'm going to. So if you were in class, I would have essentially asked you to show you one of this and asked you to do the next one. But I'm going to just skip this, and I'm going to talk to you about in general what happens when you look at the signaling pathways that are essentially part of these receptor proteins, and the looking at the secondary messengers. So look at these groups of various kinds of receptors that are there. These are all transmembrane receptors, as you can see. So you have, so you can see this, these are, you know, the cytokine receptors. So that would lead to the JAK-STAT pathway. If you were to look at, you know, survival factors, so then this will go through the PI3K pathway. If you looked at any GPCRs, then that would go to the G protein, then that gives you cyclic AMP, and so on and so forth. So you can see there is a whole host of things happening. And of course, you know, part of this is also part of the sensory system where apoptosis takes place. So this and that together will, you know, essentially mediate some kind of apoptotic signaling. So what I wanted to get across to you is a tremendous complexity, right? So so how many of you have had some kind of a background in this understanding of this thing? Yes. I think I missed out your, I closed that, and rather I should have done the other way. Hold on, just give me a second because I'm not able to see the faces here. I need to get back here. So how many of you are familiar with what I've been talking about so far? Do you recall what I said in the last class and how I'm recalling this in this class? Yes, no, what's happening?
Sort of, yes.
Yeah, sort of.
To some extent, sir.
Yes.
So what is the extent that you don't get?
Sir, the biology aspect of it is a little more comfortable.
Okay, you're comfortable with the biology aspects. What about the students who are in math and physics? See, because you see, I'm teaching this. I know that this is the students having different backgrounds. So unless you kind of interrupt me and ask me things, it's going to be a one-way thing. So difficult. But anyway, you look at, so what this is showing you essentially, it's predominantly explaining to you, you know, how the RAS components, you know, group of proteins, which are a family of proteins that encodes these small GTPases, essentially participates in this apoptotic. And the reason I put this here is because this is a runner-up to what I'm going to talk about, which is calcium signaling. So whenever we look at cells, and especially neuronal cells, we see that calcium signaling has a very important role to play. And otherwise, you see each of these groups which are there here, they are all leading to gene regulation. And there's some part of it which comes through this PKC group. If it is not NF-kappa B, then it should essentially, if it comes to NF-kappa B, then it will give you some kind of a inflammatory response. But else, otherwise, it should give you a cell cycle response. So PKC is involved in, I think, through this three groups here, which will give you a gene regulation that enables, you know, cell cycling, growth and division and other things for certain kinds of cells. Whereas here, this pathway will essentially lead to apoptosis. So there's always this balance between cell cycle control versus apoptosis. So there's some parts of it which will tell the cell you need to repair and, you know, survive, and whereas, you know, in another part it will say, well, you need to allow this disintegration to take place so that, you know, these cells can come in its place. So, but I will go in details now to look at signaling pathways, and one of the signaling pathways that are very important and critical is the calcium signaling pathway. But before I come to this, this was an example. How many of you have done this? This was supposed to be the second assignment. Because of the minor, I think I've not insisted on this, but I will be assigning this as a second assignment later. So this is a very oversimplified diagram of the insulin signaling pathway, where essentially what you look at is, you know, how this glut flow response changes with changes in insulin. And if you go back and look at how this insulin regulation takes place, then you'll find that this plays a major role in phenotype becoming diabetic. So these are things that are there, and there's much more to it than meets the eye. But the whole purpose of this assignment is for you to get down to how to do wiring diagrams and convert wiring diagrams to differential equations, and use these differential equations in Simulink, for example, and then do a coding and look at the response for given certain changes in the input condition. All right. Okay. So we look at the case study here. We're looking at calcium homeostasis. Anybody can tell me why calcium homeostasis is important? No? Yes? Again, I have to come back
So
here.
from what I know, I guess calcium is a very important like transduction molecule for even, I guess, neuronal impulses as well. So if you don't maintain like a homeostatic environment, then I keep forgetting because I studied about this a bit
I know.
a while ago.
So you're on the right track, but I was going to ask you, what about other metal ions then?
So I guess sodium and potassium also play a similar role as calcium does, but calcium homeostasis is a bit more important than sodium and potassium, but I forget the reason why.
Sir, I think calcium is something that it triggers the neurotransmitters.
Who's that now?
Naveen.
Naveen. What happened to Ratnesh? That fellow just disappeared. I was going to ask you guys what happened to Ratnesh, because so today I was just kind of going through, looking at what we had done so far, and then I realized that Ratnesh has not been even answering any questions and not being in the class. Anybody know what happened to him?
Not really. So.
Okay. Anyway, just ask him, to come back and join the classes, and in case any difficulty, to let me know. But let me just, just follow up on this question. The reason I'm just stopping here before we get into the main topic is the fact that, as rightly said by Shantan, is that sodium and potassium are well known for giving you the response for what we call, the action potential, right? It just gives you the action potential, and that moves like a pulse along a wire. And that is the primary work of the sodium-potassium channels, and the density of the channel along the axon and other things are very important, no doubt. But at the synaptic junction itself, there are other things that happen, and one part of that is based on this calcium signal that takes place. Also, the calcium that is there within the cell is primarily in two compartments. One is in the smooth endoplasmic reticulum, which is the main storehouse, and the other one is the cytoplasm. And then you have this calcium that is there on the outside of the cells, which essentially comes through the leaky channels. So you have leaky channels through which calcium can pass through. And there is a very nice balance between all of this. So that is another reason for study. But there are a host of receptors which respond differently to different concentrations of calcium. So there's a graded response, and also it changes with the presence of other ions in the system. For example, NMDA receptors are there, respond to calcium and magnesium, and how exactly is going to respond will depend on the related concentrations. And this becomes quite significant when we look at various kinds of phenomena within the cell. Why is it that this is important? Because the amount of calcium that goes in and at different kinds of requirements of the cell dictates how the milieu around, not only the cell itself, but what is around it, how it behaves. So if you were to look at just neuronal cells, then in the brain itself, we will have other kinds of neuronal cells, which are astrocytes and glial cells. So astrocytes, for example, are very sensitive to changes in calcium in the environment, and how it modulates the capillaries to allow more flow or less flow of blood in a certain region is a very well studied phenomena. And also all the fMRIs that are done is based on that. What's called the bold signal, right? Blood oxygen level differences. So that signal is what is being measured, and that comes because of the astrocytic activity that is there in the brain. And of course, the signal itself is based on oxygen levels, and that oxygen level will change only if that is modulated by calcium signal. So quite a bit is there, and so just my own experience, it's like that there are just so many links in the chain. There are so many different components that the more you read and try to study, you get to the point where you realize that any one thing can go wrong, and that could essentially trigger a disease. Whereas also the fact is that it's miraculously balanced over evolution, that it has certain amount of robustness that it doesn't go wrong so easily. So it's something that you need to ponder upon. And famously, Richard Feynman is known to have said, it's one of the quotes of Richard Feynman, that the human beings are a universe in themselves, contemplating on a universe on the outside. So we are thinking about how life and these planets and everything originated, the entire universe originated. But if you look at ourselves, we ourselves are universes in ourselves. If you just look at it from the scale of a microorganism. So anyway, so that's something to ponder about. So I'm going to go back now to the main topic again here. And there's a little bit of history. So the discovery was done somewhere around the 1883s. And the progressive work that was done was, the significant work was done in 1943 and 1947. And if you remember, this is the time when actually also we had the work which was done for neuronal cells. And calcium evolution during this time also, the difference between prokaryote and eukaryote cells, the intracellular calcium stores, all of these were well studied during this period. So, but what was studied at that point of time, even today, it's a significant amount of. So this is like the discovery part of it. But even though we know so much about it even today, as this role as secondary messengers, there's still so much to be known. And this is an example of Thioplasma sulfoxide bacteria, which possesses an endoplasmic reticulum, endoplasmic membrane structures here, and these structures are the structures where essentially you find these calcium stores, calcium ions being stored. All right. Now, so the two main contending ions are magnesium and calcium, and they are very similar even on the periodic table. And what you'll find is that this calcium has a coordination number between 6 and 12, whereas magnesium has a fixed coordination number of 6. So, which is the reason why evolution has chosen calcium to be more dominant than magnesium. So, if you look at situations where calcium and magnesium are there together, calcium will always dominate. So, there are other ions also, and in the cellular cytoplasm, which is there in intracellular regions. And so in general, we have this kind of an equation to talk about a metal and a ligand, metal ligand coordination which takes place. And of course, we all know about hemoglobin, but there are also zinc and copper ions which play a major role, and one of them is SOD1. So copper, zinc, SOD1, which is part of primarily understood from the point of view of neurodegeneration in cells. So those studies have been also extensive. It tells us quite a lot about how neurodegeneration occurs. But let's go back to calcium. And when we talk about calcium here, we look at how free, bound, and trapped calcium behave in coordination with each other. So in the cytosol, we have calcium, which is maintained at a very low level compared to sodium and potassium. It's much lower. So there are many. The reason why it still works is because the affinity for calcium binding is very high in proteins. So there is also this active transfer of calcium ATPases which are there, which enable to maintain this at a low value. So there are these enzymes that are working overtime to help maintain this condition. And we'll look at this in a minute, why that's important. Now, also, there is a specificity of this over magnesium, calcium over magnesium, and we talked about of that in terms of the coordination number of it. So there are many other mechanisms that are there, and especially in hormones. So those are the long-term effects, but you have just local binding of protein. Those are the short-term effects. So let us look at values. So calcium will be approximately in the millimoles, and this is in the extracellular plasma fluid. And when you look at intracellular resting potentials, you'll find that this is in the nanomoles. And in the ERs, you have about there's nearly the same values. So the pK values of this are given, no? The pK values which are pH. So this is the value for the P of calcium which is there inside the cell. So just like you have a pH, you have a pCa which is there, and those values are shown here. And of course, the magnesium that is there, in comparison, you can see that there in this case it's nearly the same, but in the cells much higher than that. So if you have excess calcium in the system, then you have hyperglycemia, which this is important to understand because it reduces neuromuscular transmission and it causes myocardial dysfunction. So this is one of the reasons why also you'll find that that you have calcium blockers for those who have hypertension, right? And hypocalcemia also affects excitability of membranes, and if continued can lead to tetany, seizures, and death. So this is another situation, and some of the poisons that essentially work in this fashion. They sequester out the calcium, and you have neuronal seizures, which essentially will give you problems. Seizures in general, are hallmarks of excitability of calcium, so either good or bad. And whenever there seizures occur, usually it's tracked to some locus in the brain, where you find that something in that region about calcium is not right. That means how calcium is being dealt with due to some kind of either a genetic problem or some other problem. You have a disbalance or dysfunction of this calcium homeostasis that is present there. Any questions so far?
None currently, so it's clear.
So all of you are understanding everything. So I think some people have left already, huh? Right. So they don't find interest in this. I mean, I don't know. I hope I'm doing a reasonable job in this. I wish this was all in class because some of these things are really a little dry if I can't teach it in class. Anybody okay? Everybody's okay with this so far?
Yes, sir.
Yes, sir.
Sorry, I just drank some water. Okay, let me continue. Now, there are these, this is about how calcium was essentially analyzed or studied in cells, and so it started with alizarin dye from amoeba, to radioisotopes, then to aquarins, and then to fluorescent dyes, and you had Quin2 and Fura2, which are the most recent ones. So this is mainly information which you can go back and read. I'm not going to waste time here. So essentially, this Quin and Fura essentially have this crab-like claws in which they can hold on to some calcium. So 1 calcium, 2+ would be there, bound by these negative oxygens here. So Fura2 and this one. So you can see that they have this large hydrophobic tail or group here, which enables this to go through the cell wall and allows this calcium to be sequestered. So, and based on that, you can study what is the concentration of calcium inside the cytoplasm. Okay. So mechanism that elevate calcium in concentration and so there are these primary groups which are there. So one obvious reason is that the switchable membrane channel. So whenever you had evolution go over the various kinds of metal ions which are present in cells, so you'll find that there are 4 or 5 metal ions which we dominate: zinc, copper, calcium, magnesium, and of course sodium, potassium, so which is there. So these are the primary ions that you'll find, and the reason why they are there, is because these are essentially very easily used to neutralize charge or to give charge. And so in electrically excitable cells, the Ca2 is important, and of course they will actually act through certain kinds of receptors that mediate this. So the one kind of receptor that does this is the inositol triphosphate IP3 receptor, and there's the ryanodine receptors. So these are the ones which are there, which mediate this transmission of calcium into the cells. So let's look at a typical, signal transduction pathway, where we are looking at, how calcium is essentially mediating some action at the bottom. So we talked about this earlier. So there's a ligand, there's a receptor, then you have a G protein here with alpha, beta, gamma components. And in this case, if this one is actually altering or interacting with the PCL betas, then the path that will follow is the IP3 to the IP3 receptor. And this is this phospholipase C beta, which will produce this inositol phosphate 3. It also produces the DAG component, which is, let me look at it. I keep forgetting this value here. This is diacylglycerol compound, which is here. All right. So this diacylglycerol will then act upon phosphatase kinase C, and that essentially has other series of interactions which take place to other gene regulation processes. Whereas here it's going to release calcium from some store, and then that release will essentially give you a burst of calcium signals, which is going to be there to mediate some other work within the cell. All right. So one study that I'm showing you here is essentially there to show you, how calcium signaling takes place and how this measurement takes place. So MAG-Furat2 is the fluorescence of that is shown on this axis here. And what you're doing is you're looking at cells which are the same location, but you're taking snapshots at different times with different treatments. So you're looking at the ratio of 340 to 380, which means that you impinge at 340 and you look at the response at 380, or you look at the 340 itself. Then you look at the intact cells with 1.8 molar calcium, and you don't see any change. So that's the baseline for the cell type 1 and 2. So that's what you're looking at. Then you look at intact with slightly higher amount of calcium, but you have given something called digitonin. So digitonin is essentially going to permeabilize the cells, and so the calcium just leaves, so you don't get the signal there. Now, if it is permeabilized and you don't have ATP, nothing really happens. So but the digitonin has been moved out, but the signal does not come to a level where you can essentially see anything at all. So essentially only after the ATP comes in here, like you see in this last thing, that the signal goes up, and then that signal will continue as long as the ATP is present in the presence of calcium. So that is showing you, this entire process of, how this imaging of calcium is done from calcium stores. So you can essentially see that. And actually, if there's a time lapse, so you can see these punctas here. So that shows you the stores in which this is all getting activated. So if in case you have a complete video taken of astrocytes with this kind of an experiment where you suddenly are allowing calcium to calcium signaling to arise within the cell, then you'll see that it goes in waves. So those are called the calcium waves, and we'll talk about that a little bit in some slides from now. So the patterns that you see there, of course, you have to keep in mind this temporal. That means is going to be transient in its measurement. When you measure it, there'll be a high and a low. There'll be some baseline, then from there there'll be an increase, and again a period where it'll come back to the baseline. So there is also some spatial detail, which means that if you saw the punctas, that it actually shows you that there will be variation from one location to another within the cell. So there are these so that essentially will tell you that there are some release events which takes place. And of course, the signals are electrically excitable in cells. That means you can have voltage gated phenomena that you can essentially control. And then you can have localization of secondary messengers. So which means that, because they are there, you have these stores which are there, calcium stores in the cell. There will be a local phenomena that you will have to, that you could actually use to explain how calcium essentially mediates the various kinds of phenomena that you see within the cell. Okay, so this is the part where this is shown here. And you can see there are 5 panels that are there on the top. And the 5 panels that are there are at various levels of calcium. So one is at 1 and 2 are at 50. So 1 is at 50, 2 and 3 are at 500. Sorry, and 4 and above you have this in this category you'll have along with this. So you'll have this. Sorry, 50 was at this during. So you have 500 at this level. So it's going to be the 500 continuing at this point. So the 500, of course, this spike that you see is for this 4 and 5 only. So, all right. So you can see that when you have very low levels of this carbachol, which is there, and so in a calcium-free solution, which you have this, and then you gradually increase. The calcium is gradually increased in this quantity, so you can see that there is a bath solution which is containing calcium at some levels here. Only when you are increasing this compound that you start seeing these spikes that are coming up here, which is part of a study which was done for a single smooth muscle cell. So when you increase it to 500, there's a spike that happens, and that brightness is seen here. And as it goes down, you can see that brightness goes down. But if you essentially allowed this to happen in the presence of external calcium, then what you will see is that there's a lot of oscillation that takes place because of the presence of calcium in there. Okay, so one case where you see is essentially this calcium-free solution here, and the other case you see is a calcium-containing solution here. So this oscillation that is there is something that people have tried to model and to understand why this happens. Okay, somebody has to. I'm getting not getting any feedback from the people. So
what is the role of carbachol here? I didn't really get it.
Huh? So essentially, it's
what is the role of carbachol here?
So essentially, it's going to remove the. It's essentially the solution that will essentially remove the calcium that is present. So you have calcium. Carbachol is going to be the. I think. Oh, sorry. 1 minute. I'm not very sure. I think there might be some kind of permeability. I'll have to get back to you on this, on the carbachol. Something that I have not. Okay, I'll tell you about the carbachol later. Okay. I don't have it in my notes here. Anything else?
Nothing else.
You can just look it up. Can you just look it up and tell me? Because
yes, sir.
It's just blanking out. I'm just blanking out on this right now. So can you just look it up on the net and tell me what is this? I think it may be do with the permeability of the membrane.
So from the paper that I found, it's just written that it leads to an increase in the intracellular levels of calcium.
Okay, it might be acting on the membrane inside to release the intracellular stores. Maybe that's something that is there. So, or it may be a signaling molecule that essentially tells the cell to release calcium. I'll have to look it up. Okay. Anyway, so it's a trigger to release the calcium, and so if you did that, so what's happening is when you don't have calcium in the bath, then you're releasing the calcium which is there inside the membrane, and then here you look at calcium coming in, which is the oscillation that takes place. Anyway, so this one I have to look back and tell you. All right, leave that aside. Not everything is good. At least somebody is asking me a question, even if I don't know the answer to that. So there is another phenomena that takes place, and this is called the calcium-induced calcium signaling. And what you find is that when you have a calcium receptor, and because of the calcium being released, it's going to have a positive feedback on the neighboring receptors which are there, and that is going to kind of recruit more of these receptors to essentially open up the calcium channels and allow more calcium to enter or get released from the smooth reticulum into the outside, I think. Yes. So again, there's a lot of text here, of which the idea is that these receptors are going to be populated by more of these calcium, which are going to allow more of these calcium signals from this to get out, and then recruit more so that more of this calcium goes into cytosol. So the phenomena that if you look at this carefully here, so this signaling that comes here has changes, it has an influence over this region within the endoplasmic reticulum here, and then from here it will essentially recruit more of these, and more of the calcium current will flow through, recruit more of these channels, and so on. So it goes on. So essentially, one after the other, you release more of this, and this leads to what is called the calcium wave that takes place. So it's called a calcium-induced calcium release that takes place. Okay. Now let's go to looking at. I'm going to skip this slide. Yeah. So if you went into the the Keck pathways, then the Keck pathways will show you an entire network of how this calcium is essentially modulating a signal which is helping in several other factors besides what is shown here. So what's shown here is it's going to give you it depends. If you have a lot of this calcineurin being activated here, then you'll get learning and I mean, it actually depends. So you can't really say that this is going to predominantly help in learning memory, because you also have apoptosis taking place here, and you also will have other phenomena. For example, I'll just go back here. So you have three main components here. One is the cal modeling, the other one is PKC, the other one is IP3R. And all of them are actually activated and acted upon by calcium. And this secondary messenger that is calcium, which is coming out here, can do a host of things. So look at other signaling pathways, for example, or MAPK signaling. It also helps in neuronal long-term potentiation. Besides, if it is too severe, then you get into apoptosis. So there's just one more slide, and I'm going to just take the slide and then, there's a few more. Okay, we'll stop here. There are just a few more slides here, and when I start the next class, we'll just go over a few of these slides and then come back to this. So I have not done justice to 2 slides, and I will go back and come and tell you more about those in the next class. So any questions so far on the material we've done today? Yes? No?
No, sir.
Okay, so then I'll just conclude this class and we'll meet again in the next class. All right?
Sir?
Yeah.
So this is not about today's class. Is it possible for you to upload the methionine model on Moodle?
Okay, all right. I'll see that if I can do this. Okay.
Yeah, thank you, sir.
Okay.
So one more thing regarding the submission for the models, should we just submit the model file or should we submit like snapshots?
No. You need to submit the model file also. Make sure that it runs.
Fine, sir.
Okay.
Yes, sir.
And also, when you're writing the text, you have to give snapshot and discuss.
Right, sir.
Okay. All right.
Okay. Thank you, sir.
Okay.
Thank you, sir.
Thank
Bye.
you, sir.